Welcome to this webinar on standardized media and reagents for neural stem cell research. The webinar will be divided into three main topics. I will discuss how the NeuroSphere culture system has been used to advance our understanding of neural stem cell biology and regulation, and what advantages the NeuroCult media products offers towards this goal. Finally, I will discuss the new colony-forming cell assay, which offers a more reliable and accurate method to enumerate neural stem cells and neural progenitor cells compared to the traditional NeuroSphere assay. Firstly, to begin with, a quick overview of the central nervous system. The CNS consists of three major cell types. Neurons are the functional units of the brain. Neurons transmit electrical signals and communicate with each other through the secretion of neurotransmitters. Astrocytes and oligodendrocytes, collectively called glial cells, play a more supporting role for the neurons. Oligodendrocytes are the cells which produce myelin sheaths which envelope the axons of neurons and therefore provide protection. Astrocytes have a significant role in terms of supporting neuronal growth, survival, and migration throughout the development of the mammalian brain. The adult CNS was always believed to remain relatively mitotically quiescent. This belief was enforced by the long-held dogma which maintained that no neurons could form in the adult brain. It was for this reason that the adult brain could not repair itself during injury and disease, because there was no longer any cell proliferation or stem cells. This dogma stood for many years, even though a few researchers were challenging this dogma by showing some evidence of cell proliferation in the adult brain, at least in non-mammalian adults. It was not until the early 1990s that Reynolds and Weiss were able to isolate and demonstrate that there was a rare population of cells in the adult and embryonic brain which had properties of a stem cell. They were able to demonstrate that when tissue was obtained from the adult brain and cultured in an in vitro culture system they called the NeuroSphere culture system, that there was a population of cells which were able to self-renew and give rise to more progeny. Later, they also demonstrated that these isolated cells were able to self-renew and generate large numbers of progeny over an extended period of time and maintained multi-lineage potential over an extended period of time. They were therefore able to functionally define a population of neural stem cells. It is now well known that neural stem cells are present in the embryonic and adult brain in specialized niches. In the mouse embryo, for example, neural stem cells can be isolated from tissues microdissected from the ventral mesencephalon in the midbrain and cortex and striatum in the forebrain. In the adult mouse brain, shown here, neural stem cells or progenitors are shown to be present in two regions. The first one I will discuss is the subventricular zone or SVZ region, which lies adjacent to the lateral ventricles. Neural stem cells in the SVZ region are responsible for continuously generating new interneurons in the olfactory bulb. The neural stem cells produce a pool of transient amplifying progenitors which migrate and at the same time differentiate in the rostral migratory stream before they establish themselves in the olfactory bulb as mature interneurons. A similar type of neural progenitors have also been observed in the adult human brain. The other region which remains as a site of neurogenesis in the adult brain is the subgranular zone in the hippocampal formation, more specifically in the dentate gyrus. Both the SVZ and hippocampal stem cells can produce neurons and glia in vitro and also show self-renewal. The basic technique for the neurosphere culture procedure, as described by Reynolds and Weiss, is shown here. Stem cells can be isolated from many regions of the embryonic mouse and rat brain using the neurosphere culture system. The original procedure for the neurosphere culture system is based on dissecting a tissue from the region that's being studied. The tissue then needs to be disassociated either mechanically or enzymatically in the case of adult tissues to give a single-cell suspension. The cells are then plated at a known cell density in a serum-free media which contains a mitogen, for example, epidermal growth factor or EGF. Under these conditions, the majority of the cells will die. However, a subset of cells which are responsive to EGF will proliferate to form clusters of cells called neurospheres. These neurospheres continue to increase in size over time, and after the culture period of approximately 5 to 7 days, will form neurospheres with a diameter of about 100 to 200 microns. These neurospheres can be harvested by centrifugation and the neurospheres dissociated to produce a single suspension. The cells from this suspension can then be replated at a known density in the second culture under the same media conditions as the first culture. After another 5 to 7 days, neurospheres will be generated again. This procedure can be repeated over and over again. However, the success of generating new neurospheres at each passage depends on the maintenance of neural stem cells in the culture and the ability of those stem cells to self-renew and generate large number of progeny. If neural stem cells are maintained in the neurosphere culture over time, if you graph cumulative total cell numbers obtained versus the number of passages performed, you will obtain a graph that looks like this. There is a huge increase in total cell numbers over time. The only way this type of expansion can be achieved is if there are neural stem cells present in the culture and they are self-renewing themselves and at the same time generating a large number of progeny. Therefore, by replating cells from neurospheres over several times, you can demonstrate the presence of neural stem cells because they remain undifferentiated, they self-renew and produce large numbers of progeny, therefore fulfilling 3 out of 4 of the functional criteria listed here. However, in order to prove that stem cells are maintained and retain all stem cell properties, it is necessary to also show that at any time during the neurosphere culture, intact neurospheres or dissociated cells derived from the neurospheres can be removed, the growth factor containing media removed, and the cells exposed to a low level of serum and an adhesion matrix such as poly-D lysine or poly-D ornithine, and the cells will immediately differentiate into the 3 cell types found in the mammalian brain. Shown here are immunocytochemistry images of neurons in red, astrocytes in blue, and oligodendrocytes in green, stained with antibodies against cell-specific markers. Therefore, the neurosphere culture system, when employed in this manner, i.e. long-term expansion and multilineage maintenance, the neurosphere culture can be used to functionally define neural stem cells. A neural stem cell fulfills all of the functional properties defined, and a cell that only fulfills 1 or 2 of these criteria is not a stem cell, but rather a progenitor. Here at Stem Cell, we have developed a complete set of tools for working with the neurosphere culture system. We have media for the isolation and expansion of neural stem and progenitor cells from different species, kits to prepare tissue and disassociate neurospheres, and finally, media formulations for the differentiation of neural stem and progenitor cells. While there are other commercially available media formulations which are used for culturing neurospheres, the NeuroCult product line developed by StemCell has some critical advantages which I will discuss. The media formulation developed by StemCell is closely based on the original formulation by Reynolds and Weiss. Based on the formulation, we have fine-tuned the media formulation to be specific for the species that you are working on, be it mouse, rat, or human. As I discussed in detail, it is important that any media you use has been optimized to maintain the neural stem cells in culture. Other media may allow for neurosphere formation. However, this may be limited to one or two subcultures. It is evident also when using NeuroCult that high expansion is seen in early and late passages of cells and throughout the culture period. While the Reynolds and Weiss formulation is published, we have pre-screened all ingredients and used only high-quality grade tissue culture reagents. Each batch of media is quality tested in the Neurosphere culture system and not in surrogate assays. By using high-quality and pre-screened components, we have maintained reproducibility in our media performance and quality. With this, our NeuroCult products may facilitate standardization within and between labs. The media and protocols for culturing neurospheres from embryonic and adult mouse CNS tissue are presented here. We have developed a specific formulation consisting of a basal media and a proliferation supplement. The basal and proliferation media is combined and the mitogen EGF is added. For adult mouse cells, EGF, FGF, and heparin need to be added to the media. Mouse cells cultured in the formulation will produce neurospheres in 5 to 7 days. These neurospheres can be collected and dissociated into a single cell suspension and replated in fresh NeuroCult media. At any time, the cells can be cultured in the basal medium supplemented with a differentiation supplement, and immediately the cells will differentiate into neurons, astrocytes, and oligodendrocytes. The NeuroCult kit for mouse, therefore, can be used for the expansion, differentiation, and cloning of cells in smaller well formats. Shown here is the expansion of total cells over time when E14, embryonic day 14 cortical cells are cultured for multiple passages in the NeuroCult formulation. At each passage, a 5 to 10-fold expansion in total cell numbers is obtained. The viability of cells is also maintained at around 80%. The NeuroCult media also supports the long-term expansion of cells isolated from adult SPZ region. These cells are technically more challenging to culture in the neurosphere culture because oftentimes new neurospheres are difficult to generate at each passage. Here, in the NeuroCult media formulation, adult mouse cells expand significantly over time with a 3 to 10-fold expansion seen at each passage. There are common problems associated with culturing rat neural stem and progenitor cells. Often, the rat cells are sticky and tend to adhere to the bottom of the well or flasks. When this occurs, it is difficult to generate new neurospheres from the dissociated cells. Cells will tend to stick down again at the next passage and over time. Neural stem cells are not maintained and no expansion in total cells is obtained. By developing a new media formulation and protocols specific for rat CNS cells, we have been able to overcome some of the problems associated with culturing neurospheres from the rat CNS. And we have been able to demonstrate long-term cultures with these cells. A specialized basal and proliferation media formulation have been developed for rat cells. The procedure for generating neurospheres from rat CNS cells is similar to that for mouse cells with some key differences. The media for rat cells is species-specific, and you have to add EGF, FGF, and heparin to the media. Neurospheres are cultured for only 4 days compared to 7 days for mouse cells, and it is very important that a half media change is performed during the 4 days. Neurospheres of about 100 microns are formed by 4 days, and these have to be passaged. The neurospheres are mechanically dissociated, and the cells replated in the same media conditions to generate new neurospheres. At any time, the neurospheres can be removed, the growth factor containing media washed away, and the cells resuspended in rat basal medium and differentiation supplements in order to induce differentiation. Here we compared the NeuroCult rat media to a traditional formulation. We cultured the rat cells in either T25 centimeters squared flask or in ultra-low adherent wells as recommended by the traditional media supplier. In green, you can see the significant expansion in total cell numbers over time for rat cells cultured in the NeuroCult formulation. In the traditional formulation, cells were not able to form new neurospheres beyond passage 3, and we lost these cultures. In the ultra-low adherent wells, again, the expansion of total rat cells in the NeuroCult media was higher compared to the traditional formulation. However, the overall expansion was reduced compared to culturing in T25 flasks. These results indicate that the type of plasticware and media is very important to maintain healthy neurosphere cultures from embryonic rat CNS cells. And the recommended conditions include using the NeuroCult rat formulation in T25 flasks. We have also developed a specialized basal and proliferation media formulation for the long-term culture of fetal human CNS cells. Here you see the growth curves obtained when fetal telencephalon and cortical cells were cultured as neurospheres in the NeuroCult media formulation, showing expansion in total cell numbers over time. The same NeuroCult media for the culture of human CNS cells can be used to culture tumor CNS cells. Shown here is the expansion curve for human glioblastoma multiforme cells, or GBM cells, cultured over multiple passages in the NeuroCult media supplemented with EGF, FGF, and heparin. As you can see, the growth curve for the human GBM cells is inhibited by BMP4, and you see a decline in expansion over time. Therefore, the NeuroCult media can be used to culture GBM cells, but also to look at the effects of different cytokines on the growth of these tumor-derived cells. We have also developed a xeno-free version of the NeuroCult media, or an animal component-free media. The advantage of this media formulation is that it does not contain any components which are derived from animal, and therefore it would be considered a safer, more compliant media formulation. If you have plans to move your research to preclinical applications, this would be a good media formulation to use to begin those types of studies. Here is the growth curve generated in experiments, 3 experiments where fetal human CNS cells were cultured in animal-containing media or in the ACF media, showing similar expansion in total cell numbers when either formulation was used. As important as the media to culture cells, other supporting tools, such as the tools to process CNS tissues or neurospheres, are equally important. I will discuss some of those tools which were developed to dissociate neurospheres and tissues. One of the big technical challenges in the neurosphere culture system is to determine when the neurospheres are ready to dissociate and replate in the new media. Leaving the neurospheres to grow too large causes problems because the center of the neurospheres become necrotic due to the lack of oxygen exchange. Passaging the neurospheres too early will give you too few cells for the experiments and compromise cell proliferation. One way to assess neurosphere size is to take an aliquot of the culture and observe it directly under a hemocytometer. If you drew a line through two grid lines, this is approximately 100 microns. You should passage the cells when the spheres are between 100 to 200 microns, as seen here. However, nothing replaces the trained and experienced eye of the researcher. The top panel shows neurospheres that have been left too long. The neurospheres are dark and have very irregular periphery and some cells blebbing off from the periphery. In the bottom, the neurospheres are intact. Phase contrast bright and have nice round outside perimeters with healthy cells on the edges, and these cells are not breaking off. These neurospheres are ready to passage. We have developed a chemical dissociation kit which uses no enzymes or mechanical procedure. The procedure is outlined here. The neurospheres are collected by centrifugation, the supernatant is removed, and the pellet is left. You would then add solution A to resuspend the pellet, followed by solution B, at which time you will start the timer for 8 minutes. At approximately 3 and 7 minutes, the cell suspension is gently mixed. At 8 minutes, the final chemical solution is added. The entire suspension is then resuspended in the NeuroCult media or any other buffer for further analysis for cell counts and viability counts. Importantly, when mouse cells were treated at each passage with the chemical dissociation procedure and compared to cells that were treated with the mechanical procedure, which is a more harsh method of dissociating neurospheres, the viability of cells treated with the chemical dissociation kit was significantly higher. This higher viability also allowed for better expansion in total cell numbers in long-term cultures, presumably because there were more viable cells in each subculture. Acutase is a cocktail of enzymes that works well to dissociate mouse neurospheres in long-term cultures because the stem cells retain their stem cell properties. Shown here is the expansion curve for cells treated at each passage with acutase in orange versus the mechanical procedure. As you can see, the total cell expansion and viability is higher in the acutase-treated mouse cells. We have also developed a kit to prepare tissues obtained from the adult mouse or even fetal human tissue. The adult tissue requires enzymatic procedures to obtain a clean single-cell suspension. The SVZ region is dissected from up to 8 adult mouse brains and collected in the specially formulated collection solution. Once all the tissue is pooled, the collection solution is removed and the tissue is quickly minced with a scalpel. Next, the dissociation solution is added and the mixture pipetted. The tissue is incubated for 7 minutes at 37 degrees. After 7 minutes, the inhibition solution is added and the mixture gently pipetted. It is important to avoid adding or creating any air bubbles, which will cause cell death. The cells are then washed 3 times and the final pellet is resuspended in an appropriate media for cell counts. We use the kit for all adult-derived tissues. Finally, we have developed specific kits for the differentiation of mouse, rat, and human cells. Intact neurospheres or single cells dissociated from the neurospheres are plated in the differentiation media in the presence of a matrix like poly-D ornithine. The cells will immediately attach to the substrate and differentiate. The mature cell type can then be detected with primary antibodies against cell-specific markers. Shown here are neurons detected with an antibody against MAP2, astrocytes detected with an antibody against GFAP, and neurons detected with an antibody against beta-tubulin. And finally, oligodendrocytes were detected with an antibody against myelin basic protein, or MBP. At Stem Cell, we supply all of these cell-specific antibodies as well as the secondary antibodies. Shown here are rat cells cultured as neurospheres for 10 passages and dissociated into a single-cell suspension and replated in the differentiation media for rat. A triple-stain immunocytochemistry was performed on the differentiated cells, and the number of neurons, oligodendrocytes, and astrocytes were counted. As you can see, neurons make up 2 to 5% of the population, oligos around 5% of the differentiated cell population, and finally, the majority of the differentiated cells are astrocytes. To summarize this section of the webinar, the advantages of the NeuroCult media and reagents are that these products have been developed to be used to study neural stem cells based on a functional criteria in vitro. The products are designed to maintain cells in an undifferentiated state, to show self-renewal, and to expand progeny over an extended period of time. These reagents are also designed to determine if multi-lineage potential is maintained over time. Our next step was also to develop a new tool to enumerate neural stem cells and neural progenitor cells, a tool which we believe did not exist in the field up till now. The NeuroSphere assay has been widely used as an assay to measure the number of neural stem cells, either by simply measuring the ability to form neurospheres, secondary neurospheres, or the formation of neurospheres as an indicator of stem cells. However, a major limitation of the NeuroSphere assay as an assay to measure neural stem cell numbers is that not all cells capable of forming a neurosphere is a stem cell, in that cells that can form neurospheres that do not fulfill the functional criteria of a neural stem cells. A cell that forms a neurosphere may not be able to self-renew, produce large number of progeny over an extended period of time, or maintain multi-lineage potential over an extended period of time. In other words, a progenitor can also form a neurosphere as well as a neural stem cell. We tested this experimentally, and the experiment is represented here. We selected 34 individual neurospheres and dissociated each of those neurospheres separately and plated the cells from each of the individual neurospheres into individual wells of a 96-well plate. We waited to determine if new neurospheres were generated from the cells from the individual neurospheres. If new neurospheres were generated, we dissociated the new neurospheres and replated all the cells from that population into a new 96 well, and continued to do this as long as we could obtain new neurospheres in the subsequent culture. Each culture passage was counted. Out of the 34 individual neurospheres we picked, only 2 contained cells which continued to give rise to new neurospheres at each passage. The majority of the neurospheres contained cells which could only generate neurospheres for 3 or less number of passages. This result indicated that only 2 out of the 34 neurospheres potentially contained cells which had high proliferative potential. Another problem with using neurosphere number as a measure of neural stem cell numbers is that neurospheres tend to aggregate or fuse because the neurospheres are floating in suspension. When cells are plated at a very low or low plating density, fusion of neurospheres happens very frequently, making it difficult to distinguish if a neurosphere is made up of two or more neurospheres attached to each other. Because of these limitations of the neurosphere assay, in that one of the major limitations is that only a subset of neurosphere-forming cells are stem cells, sphere number is not an accurate measure of neural stem cell frequency. And if neurosphere number is used as a readout for neural stem cell frequency, it would lead to the conclusion that the neurosphere assay overestimates the number of neural stem cells. It is for this reason that we developed the NCFC assay. The NCFC assay procedure is outlined here. Cells from the mouse cortex or cells obtained from the neurosphere culture can be used in the NCFC assay. A single cell suspension is obtained from these tissue sources and filtered through a filter to remove cell aggregates. The cells are mixed with the NCFC serum-free medium, supplements, and the cytokines of interest. Collagen solution is then mixed in with the cells, and the entire mixture is plated in 35 millimeter dishes in duplicates and incubated for 3 weeks. At the end of 3 weeks, or 21 days, colonies are scored. In the NCFC assay, we plated the cells at a very low plating density to ensure that a single cell would give rise to a single colony in the collagen matrix. We used semi-solid media, collagen, to prevent cells from moving or aggregating. The question we asked was, during this prolonged incubation of 21 days, would some cells give rise to small-sized colonies, others to medium-sized colonies, while yet others would give rise to very large-sized colonies? The hypothesis was, can colony size be used as an indicator of the proliferative potential of the original colony-forming cell? Therefore, a cell with high proliferative potential will form a large colony, while a cell with low proliferative potential will form smaller-sized colonies. This was in fact what we observed after 21 days of culture. In many experiments, we saw colonies of different sizes. We categorized the colonies into 4 size categories: those which were larger than 2 millimeters in diameter, colonies 1 to 2 millimeter in diameter, colonies which were 0.5 to 1 millimeter in diameter, and finally small colonies which were smaller than 0.5 millimeters in diameter. We observed colonies which fit into these size categories in many experiments. We next validated our question, which is, can colony size be a measure of proliferation potential of the colony-forming cell? To do this, we cut out individual colonies from each size category, and we did so for many colonies of the different size ranges. We digested the collagen matrix with collagenase in order to obtain all the cells from each colony that we harvested. We plated all the cells from each individual colony into individual wells in a 96-well plate in neurosphere culture conditions. Therefore, all the cells from one colony will go into one 96 well. We performed this for between 50 to 100 individual colonies. If the cells from the colonies formed neurospheres, the neurospheres were dissociated and the cells replated in subsequent neurosphere culture media. As long as new neurospheres were formed from the cells of a colony, we kept replating them to determine if new neurospheres were formed. At different stages, we took some of the cells from the neurospheres and differentiated them. What we found was only the cells obtained from colonies greater than 2 millimeter in diameter contained cells which were capable of forming new neurospheres upon serial passaging. Shown here are 6 representative colonies greater than 2 millimeter in diameter isolated from adult or embryonic mouse CNS cells. The cells from the smaller size colonies could not produce long-term cultures. This indicated that the large colonies, greater than 2 mm in diameter, contained cells with high proliferative potential. When cells from the different size colonies were differentiated at either early passage or after passaging the neurospheres that were generated from the cells of the colonies for at least 10 passages, only cells from colonies greater than 2 mm in diameter produced neurons, astrocytes, and oligodendrocytes. All the other colonies contained cells which could only form two cell types, indicating that only the colonies greater than 2 mm in diameter contained cells which maintained multi-lineage potential over time. Therefore, we have developed a single-step assay that is able to discriminate neural stem cells from progenitor cells. Cells from colonies greater than 2 mm in diameter meet the criteria of a neural stem cell, and cells from colonies smaller than 2 mm are likely derived from progenitors. So, what is the NCFC useful for? For example, the NCFC assay can be used to measure the frequency of neural stem cells in a sample of cells from the SBZ region. If SBZ cells are plated in the neurosphere culture and the NCFC assay, you would only obtain the number of neurospheres formed as a quantitative measure. However, in the NCFC assay, by looking at the number of colonies in the different size categories, you would be able to determine that only 0.02% of the cells plated from the SBZ sample formed colonies greater than 2 mm in diameter, which indicates that only 0.02% of the cells plated are actually stem cells. We can also look at the effects of in vivo administration of EGF in the SBZ region of the adult mouse. After EGF was infused in the SBZ region, the cells were isolated and plated in the NCFC assay. From this experiment, it is evident that the infusion of EGF directly into the lateral ventricles has a significant effect on the proliferation of progenitors, as seen in the increased number of colonies smaller than 0.5 mm in diameter, and not a significant effect on the colonies greater than 2 mm in diameter, indicating that the effect of EGF was on the progenitors and not so much the stem cells. We have also used the NCFC assay to assess how well we have enriched for neural stem cells based on positive immunomagnetic selection with prominent or CD133 in embryonic mouse cortex. Cells were labeled with anti-prominent antibody and then without immunomagnetic labeling reagents and separated in the EasySep column-free magnet. We found that even though we enriched for up to 18-fold prominent positive cells, there was no increase in the number of neural stem cells in the prominent positive fraction compared to the negative fraction or unseparated cells. This indicated that prominent positive selection was not efficient at enriching for neural stem cells. In summary, the neurosphere culture system is a good system for the isolation and expansion of neural stem and progenitor cells. The neurosphere culture provides a renewable source of neural progenitor cells. The NCFC assay, on the other hand, is a quantitative readout for neural stem cells and neural progenitor cells, because this assay is able to discriminate between neural stem cells and progenitor cells, unlike the NeuroSphere assay. I have shown you the NeuroCult product range, which can help you in many steps used in the NeuroSphere culture system, including optimized media for expansion, kits for processing tissues and neurospheres, and differentiation media formulations for neural precursor cells. We have also added another useful tool in this field, which is the NCFC assay for the more accurate measure of neural stem and progenitor frequency. By providing a complete set of very high-quality tools for studying neural stem cells, we believe that this is one step in the path towards standardization and possibly future clinical applications of these very important cells. The use of neural stem cells in curing neurodegenerative diseases include the use of neural stem cells for treating Parkinson's disease, demyelinating disorders such as multiple sclerosis, and spinal cord injury. And finally, I would like to acknowledge my colleagues and many collaborators who have contributed significantly to many aspects of product development as well as scientific discussions. Thank you very much.